Ah, hello.
Hi, how are you?
I haven't seen you for a while.
Yeah, I haven't seen you for a couple weeks.
So all I've been doing is looking up references. And then my computer died, so all of the references died with it. So
Uh,
I did print out the important ones. So I have a printout, but I have to relook them up. So,
yeah. Use a
yeah.
reference manager
Um, no,
or? Oh, okay.
I, I just, if they're important, I print them up because I have trouble reading them online or on the computer. Like it just bothers me. So the ones I'm going to use, I print.
Oh, okay.
I've got one called ultrasound induced strain cytoskeleton rearrangement,
Oh, yeah.
an experimental and simulation study.
Yeah.
That's important because Dr. Sharif wanted me to use ultrasound to manipulate the eggs, um, and I was not sure about it. I said I don't think that's a good idea.
Right.
Well, that kind of proves my point.
Yeah.
Yeah, and not only does it heat things up, but it rearranges the cytoskeleton.
Right. Yeah, it's a
And
stress.
uh, oh, there's a few things here. They've got revealing non-trivial information structures in a aneural biological tissues via functional connectivity. Hmm. I don't know what. I don't exactly know what that's about. That's probably why I got copied. It looks like the networks.
Oh,
This
okay.
is a net. This is a networks paper.
Okay. Yeah, it sounds like it.
Are you interested in it? I can type it in the.
Yeah. Yeah, why don't you type it in?
Okay. Well, there's a couple of them here that are interesting.
All right. This one. Yeah, Blackiston. Yeah, I found that one.
Yeah, that's it.
Okay. All right. Yeah, let's take a look at it here. Right. So this is it here. Revealing non-trivial information structures in aneural biological tissues via functional connectivity. So this is uh, by Doug Blackiston, who's from Tufts at the Allen Discovery Center,
Oh,
and works with
that's
Michael Levin. It also has Josh Bongard on it, Olaf Sporns, where Josh Bongard is doing things with embodied robotics, and then Olaf Sporns is a networks person. Thomas Varley is an information theory person, and so on. Uh, so I think I actually reviewed this paper for a journal. It's okay. It's in PLOS Computational Biology. It might not be this paper, but like a similar paper. So it's my disclosure on this.
okay.
Yeah. So let's see. Okay. So yeah, this is the paper here. This is the bioRxiv version. It's also in PLOS Computational Biology. Um, so let's go in a little bit. Okay. So, so it says a central challenge in the progression of a variety of open questions in biology, such as morphogenesis, wound healing, and development, is learning from empirical data how information is integrated to support tissue level function and behavior. So you have, like, I guess, cells or something below the tissue level of organization. There's information in there, and then there's an integration of information to support like the formation of tissues. So the implication is that the cells or the things below the tissue level form a network, and there's this integration of information in the network that supports the formation of tissues. So this is there's this idea of integrated information in neuroscience that I think they're drawing from for this. But they're going to use information theoretic approach. They're going to use an information theoretic approach to provide a quantitative framework for extracting patterns from data. And so this is something that's been done in neuronal systems at the tissue level. What they're showing here is how time series of calcium ion dynamics can be used to identify the structure and information dynamics of other biological tissues. So they're interested in ion channel flux in non-neuronal cells. So, we can look at that in neuronal cells and we can evaluate action potentials and things like that. And other cells will produce potentials, electrical potentials. They're not action potentials, but nevertheless, they
You
have
hear
these
like
calcium
when
dynamics.
you still have that actin myosin complex and it runs on calcium and ATP. It's in every cell.
Right. Yeah. Yeah. So the dynamics look differently than they do in the neuron, but they're still
Hmm.
useful. Okay.
Yeah.
Yeah. So they use a calcium reporter in an organoid system, and they do this in an explanted amphibian epidermis uh, derived from Xenopus laevis. They image the calcium activity pre and post, and this is a puncture injury is the perturbation. So pre-puncture injury, post-puncture injury, and they do this for 6 replicate organoids. So they're trying to induce regeneration in that tissue. We constructed functional connectivity networks by computing mutual information between cells from time series derived using medical imaging techniques to track intracellular calcium. We analyze network properties, including degree distribution, spatial embedding, and modular structure. So these are all measurements of networks and their sort of structure. So degree distribution is the size of the network or how far one node is from another. Spatial embedding is a spatial measure, and then modular structure is how modular or how sort of compartmentalized the network is. We find organoid networks exhibit more connectivity than null models, with high degree hubs, mesoscale community structure with spatial clustering. So this is where basically it's not random, it's non-random. And we have this sort of community structure, which means that you have these groups that exist within the network, and they represent things like, different parts of a tissue or different cell types that contribute to the formation of a tissue. So there's a spatial clustering, there's community structure, meaning that there are sort of these subgroups in the network, and that relates to the sort of its ability to form tissue. Utilizing functional connectivity networks, we show that tissue retains non-random features after injury, displays long-range correlations in structure, and non-trivial clustering that is not necessarily spatially dependent. So this
So,
is, go ahead.
yeah, so that a non-injured one compared to an injured one are different network-wise, or
Yeah, the network changes its properties when you injure it, or post-injury versus pre-injury.
interesting.
So you're you're puncturing it, you're puncturing the tissue. It's changing the connectivity because the cells reorganize. I mean, if you've ever seen a video of like an injury to some, if you've seen wound healing, where the cells will sort of migrate towards the site of injury and kind of fill in, and then restructure reorient themselves to one another and restructure. There's like a fiber network that forms over the site of injury, and then they reorient themselves in that way. So it's basically changing the topology of the network.
Okay, well, that might have something to do with development.
Yeah, yeah. Well, and these aren't organoids, so you've got organoids that are I don't know how integrated they are in terms of the different tissue types, but you have basically these explants or organoids. They're growing in a culture, and if you take a cell culture and you if you take like a confluent cell culture, which means the cells are all kind of packed
Okay,
together, and you scrape it, the cells will migrate in to fill that scrape. So that's probably what's happening here. They're just kind of migrating inward to fill in the hole.
okay,
I mean,
all right.
it's yeah. Um, okay. So that's okay. Our results suggest increased integration after injury, possible cellular coordination in response to injury, and some type of generative structure of the anatomy, which I guess means that there's like some sort of like way that it sort of knows the anatomy or knows the anatomical shape or knows the anatomical structure so that it reorganizes around that structure. So again, if you're in a culture, if you put a hole in that layer or you put a hole in the structure, it's an organoid, it'll fill in that gap. It won't just kind of migrate outward and just forget about the hole. It'll fill in that hole. And so that's what they mean by generative structure. Um, while we study calcium dynamics in xenophous epidermal cells, our computational approach and analyses highlight how methods developed to analyze functional connectivity in neuronal tissues and how that can be generalized to any tissue and fluorescent signal type. Our framework therefore provides a bridge between neuroscience and more basal nodes of information processing.
Okay, well, it all sounds nice, like, interesting.
Yeah.
And
So
by
yeah.
the way, the pictures in my background here are actually a lot of them are mine. Not this side, the other side here.
Oh yeah, over there.
These are my pictures.
Great. Yeah. Yeah,
So
your
that's the other
paintings. Yeah.
thing.
Great.
Anyway, um, I do science and art.
Yeah. So yeah, they talk a little bit about information and information theoretic approaches and how this is um, kind of key to understanding collective decision making in neural networks, but you can also use this in cellular networks to look at how these structures kind of reform themselves. So, like, you think about like the example I just gave of the monolayer that's been punctured and then cells filling in to that void. They're doing collective decision making to say, this is where we need to be, and this is the network topology we need to conform to. I'm not ascribing consciousness to them, but you get my point that there's basically an arrangement or a signal that's sent that allows for that to happen. And so there are these long range correlations, and there's also a structure of phase transitions in networks of neurons that result from the network structure. And so they're now applying this to non-neuronal networks, and that's kind of where they're going with this. So in multicellular systems, cells must collectively coordinate their actions to regulate the diverse range of processes essential to multicellular life. These include regulation of pattern formation and development, morphogenesis, wound healing, regeneration, and behavior. Communication and information sharing can even extend beyond species specific boundaries, as is the case for plant animal interactions, and in symbiotic associations like lichen, where multiple species are in direct coordinated communication. Traditional techniques for characterizing coordination in non neuronal cellular tissues, thus far, include microelectrode arrays, planar cell polarity analysis, physiological reporter dyes, immunohistochemistry, and RNA-seq. So you can use all of these to sort of understand cellular function and like things like neural voltages, alignment with other cells, distributions of biomarkers, and different receptor interactions. And so what we want to do is we want to capture a longer time scale and a wider spatial scale. And so that's where they're kind of, their methods are kind of, they need to go to capture this, what they call functional connectivity. So in neuroimaging, functional connectivity is what they use to look at, like if they take images of the brain and they have, say, like fMRI data where they have voxels, and they take those voxels and they look at their activity, and they're able to correlate activity in such a way as to sort of approximate the functional connectivity. So this is where you have, if there's some activity, the voxel has a certain activation strength, and if different voxels in the image of the same activation strength at the same time, or similar activation strength at the same time, it's said that they're connected functionally. Because they're both active at the same time, it's assumed that they're doing something functionally across that network.
Okay, because I know in Xenopus tail, the cells are separated when it's developing, and so they're not one cohesive unit. So I wondered what that might look like. So
yeah,
I want to go to their lab.
Yeah.
Try it out.
Yeah. Well,
Anyways.
in this case, the organoids, I think the cells are pretty much, they have some sort of, physical connection because they're growing from an explant. Yeah.
Yeah. Well, if they're mechanically connected, that they would move together. Or you wouldn't be able to tell when one started and one ended.
Yeah. Yeah, I don't know. Well, they're using markers that are mechanical markers, but of course, then that means that they have, they share connections that are not just functional connections, but they share these sort of, in neuroimaging, they call it effective connectivity, where there's like, because obviously you have, different neurons that can be connected in the network through like, signaling, but you also have sort of physical connections or physical constraints. And so that's a different kind of network. So you can work those two networks out. And so in this case, you'd have like a mechanical network.
It would be nice to see both of them to see how they interact.
Yeah.
I don't know how you do that.
Yeah. Well, I think here, they're using like different markers, and they're able to sort of approximate the functional aspects of it, and then I guess the physical connections are kind of assumed from just them being in a common tissue, which isn't really sophisticated, right? You'd want to have some sort of map of to what degree they're connected, what degree their physical forces smushing them together or pulling them apart, and that'd probably affect their function as well. But this is information theory. So they're talking about these functional networks and how we can quantify different they're correlative maps. So there's correlated behavior, like I said, between, say, the voxel example. Even if two voxels are spatially separated, they can be correlated, and so that builds us our network. Information theory then provides a set of tools to quantify these correlations, and you can use measures like mutual information and transfer entropy to construct these networks. So mutual information is where if we take two of these nodes, we want to look at their information over time, or the information that they share in common over time. So, if you think about like statistical relationships, a good way to kind of an analogy to mutual information, or another way to measure mutual information, is to look at covariance. So two voxels or nodes will covary over time. And that covariance just means that they share variance. They share common variance. And so, in information, it's a little bit different measure, but it has to do with how much information they share, how much information, overlapping information do they share. And so that just tells us, like, if two nodes are correlated, that's great. But they also maybe are active at the same times and inactive at the same times over time. If we look over like a longer time window than just kind of one instance, because we can get a correlation for a single instance and say those two things are connected. But if we look at their time series and we say, okay, these things are active and then they're interactive and this always happens together, they share information. And so that connection means that there's some sort of information being exchanged. And so you have mutual information to measure that. And so, and I'm not really familiar with how they're applying transfer entropy, but it's going to be a similar thing where you're looking at two nodes, two, in this case, voxels, where you have this, you can tease out this relationship and make it a bit more solid. So this is where we have this, so we're looking across temporally sampled data, and we're constructing these functional connectivity networks that capture nonlinearities in structure, not apparent in static images. So they want to capture
Mm-hmm.
the dynamics here.
Yeah, too bad they couldn't do that for the surface of a xenopus egg or something,
Yeah. Yeah.
because it self perturbs itself.
Right. Okay, so a predecessor and contemporary approach to constructing functional connectivity networks are anatomical connectivity maps, which focus on physical tracts that can reveal direct anatomical links between different physical regions in a tissue. So these are these anatomical connectivity maps where they look at, like, um, these different structures in the anatomy, and look to see if there are different types of anatomical links that are functional. So this is not a xenopus egg, but it's kind of like just looking at the tissue structure and seeing where they kind of things are exchanged or things are connected. Um, However, these do not capture the long-range temporally correlated structures. So, the anatomical connectivity maps are just kind of like the structure of how anatomy is linked across, different regions. So, like in a brain, you might have long fiber tracts that go from part of the brain to the other. And that implies that there's some connectivity there. We don't know if it's functional, and we don't know what that functional connectivity looks like, but there's a physical connection there. And so that
Mhm.
physical connection then is, we can draw from that to make inferences about the functional connectivity. Um, but again, this is, um, they don't capture this temporally correlated structuredness of the tissue. So, like, we can look at a snapshot of the tissue and say this is the way it looks, but in development, the anatomy can change quite a bit. And so those,
Yeah,
yeah,
so it needs to be a time lapse.
right, yeah. And then you need to create these dynamic maps that show this, like, because you'll have structures that transform over time. So those physical maps will change.
Yeah. Oh, well, that sounds like it was a worthwhile paper then.
Yeah, yeah. So I
Not
think,
sure how I can incorporate that into anything, but
yeah. I mean, it just looks like they're doing they just do the organoid examples. They have a 4-cell embryo picture here.
and
Um,
then they take it apart, and you're
right.
going, why would they do that? Like it's a perfectly good system just as an embryo.
Yeah, yeah. So I mean, this is like where they show the pre-puncture and post-puncture data.
Oh, like, so that doesn't, like, why don't they leave it alone and just measure it as an embryo system?
Yeah, I don't know. That that might be a good baseline. I don't know if they have. Maybe they do have it in like their controls, but um, it's hard to.
Well, it changes if you excise it. Like, it will no longer. It might develop into something, or it might not. Like, it changes it.
Right, right.
So
If you take it out of the embryo context, you mean? Like,
yeah,
yeah,
it might develop into a xenopus egg after a bit,
yeah,
maybe.
yeah, yeah. And then they're just showing here where they're segmenting the tissue, segmenting the cells, and then building a network out of them. So, yeah. And then this is just more data from their time series, so they have things in time. Um, yeah, and then the degree distributions, which are um, sort of showing the nonlinearities in the network. So you have these networks that don't exchange. There's like this because there's structure in the network. There are these long tails where certain nodes serve as hubs and other nodes serve as sort of the peripheral nodes, and information transfer within those networks as a result tend to be nonlinear. That not those nonlinearities sometimes express themselves as phase transitions. Sometimes it expresses itself in terms of hierarchical structure of information processing. And then these graphs show like the degree distribution of that network. So you can see kind of how this network is organized by doing these plots. So the plots are just basically the degree versus the number of nodes. And then you can see these long tails emerge here.
Long tails are usually associated with chaos theory.
Sometimes, yeah. Yeah, if you have really long tails, networks tend to be, or systems tend to be chaotic. Yeah.
Okay.
So,
Which
yeah,
is true of a living system. So,
yeah. So, yeah. And then, of course, I do some statistical tests, the Kolmogorov Smirnov test, which is a good test for looking at long tails and how those are distributed. Okay, yeah, so this is a good paper. Again, like I said, this group, they've been doing a number of papers on looking at information processing in the cellular systems and then comparing it to like, or making the analogy, anyways, to neuroimaging. So the neuroimaging analogy is like we can build these brain networks and that's information processing, and we can say things like you could use the analogy of fMRI because it's easy to understand. And then you know so the same thing probably exists in tissues as like these, you can sample them as voxels or cells or whatever, and those nodes are connected in the same ways. The same principles hold. You have functional connectivity, effective connectivity, and you can measure them in the same way using statistics and information theory.
okay.
Yeah.
And my statistics is pretty dismal. Anyways, yeah, I got myself a primer on statistics because it's a weak point in my math.
Yeah.
So I don't know if I get to do that, but I have good Cole's notes on it. Or oh, I don't know. I could show you it. It's anyway. It's intermediate stats, and the professor I'm working with on optical coherence tomography uses statistics in all of his equations. I swear, in his signal analysis. Yeah.
Yeah.
So he's throwing statistics out at me, and I'm going, okay, then more math. I don't understand it, but I just go with the flow.
Yeah.
It's bad. Yeah. No, I've had things thrown at me like that throughout my engineering career and education, and I have no clue what they're talking about. And usually, what happens is I take the course twice, and then I can answer the questions on an exam, and I still don't understand the background. It's interesting.
Yeah.
Yeah, it's just, yeah. I don't like it. I do like to know the theory and then how that's applied. Like, it gives me a deeper understanding of things, but in engineering, they just say, know this, plunk. It's from somewhere.
Yeah.
If you had time to look it up, which you usually don't.
Yeah.
Yeah. Oh, well, I could try to find that other paper. I've got one here, but it looks like an older paper.
Oh, yeah. Which one is that?
Oh,
Yeah.
and here's one: organization of embryonic morphogenesis via mechanical information.
Okay.
Does
Sounds
that sound
interesting.
interesting?
Yeah. Yeah.
It's 2019. Oh yeah, that's the second one I put there. Um,
Oh, this one. Okay.
yeah.
Let me pull it
Oh,
up
this
here.
is a zebrafish, and the zebrafish tails are they're the ones with the proven loose cells, and they think that it elongates partly because of the cells are not as packed in the tail.
Okay. Yeah. So this is the paper here. Organization of embryonic morphogenesis via mechanical information. This is so the highlights here: positive feedback between BMP signaling and Eve1 functions in the
Yeah.
tail organizer.
Okay, I don't know what Eve one is.
It's just, yeah, it's some signaling molecule. So they set up this signaling cascade. Perturbation of organizer signaling is long range effects on cell motion. So you have this organizer signaling. If you perturb it, you can affect the motility of cells. These long range effects are beyond the range of organizer signaling, and these long range effects are mediated by mechanical information.
Mm-hmm.
So I think this figure shows kind of what they're talking about, where you have the anterior and posterior end, you have the posterior neural tube in purple, and then the dorsal medial zone here. And this is where you have this coordinated and directed cell movement down, axial movement down from anterior to posterior.
And then
So
there's this swirl there, and I think that's the eye, right? Eye.
yeah. So, well,
No,
this is the
placode.
tail. No, this is the tail organizer.
Oh,
This
okay.
is
So
where,
it's a placode of some sort. Is that?
yeah, I think so. So it's like the cells are moving down this way to the posterior end. There's a tail organizer at the tail. There's no
Oh,
tail yet, but it's going to form the tail. It's like, kind of setting up the signaling coordinate system for the tail. It says here's where
okay.
the tail should
All
be.
right. It's is that swirl an actual movement then?
Um,
Yes.
I think it's just like showing that it's moving around that space. I don't know
Oh,
why
okay.
they put
It's
a, yeah. I don't
okay.
think
So
it's,
it's an attractor or an organizer. They're calling
yeah,
it.
yeah. I guess you could say it's an attractor, not necessarily in the mathematical sense, but yeah. Um, and so you have anterior to posterior movement. The cells are coming in as raw material for the tail. The organizer is telling it where to go. Um, there's this rapid, less coordinated cell movement, so it's not moving up and down the down the axis, but it's kind of sticking around the tail area, and then
Okay.
it's thickening out the tail. So it slows down the migration. And so you can see where you have this system that if you perturb it, it will affect the movement of cells. It will kind of extinguish the potential to form a tail. And then, of course, when you have all these cells down here being organized in the tail, there are all these mechanical interactions that then accentuate the formation of the tail at the posterior end. So this
Okay.
is,
That's interesting.
yeah. And so these images are showing the organizer. I guess this is at the posterior end. So you have BMP4 and EVE1. They're kind of co-located, as you can see here. They break them out here, but these are where they're together. And then you have DMSO, which is the perturbation, DMH1, showing that, when you put that treatment there, you can obliterate the expression of these signaling molecules and it extinguishes the organizer. So that's that image. Going back to the paper, the tail organizer is located in the posterior tail bud, and both the mesoderm and epidermis express the secreted signaling proteins BMP4 and BMP2B and the transcription factor EVE1. And so this BMP signaling at the tail organizer is required for the zebrafish body elongation, which you'll see in the formation of the tail. And so then there are these different BMP inhibitors that occur as well. These are just kind of, restricting the way that, because when you express BMP, the signal kind of covers a wide region, and then you have an inhibition in places where there shouldn't be a tail. So if you got rid of the BMP inhibitors, the tail would not be as well formed, let's just say. So you have these BMP inhibitors, chordin and noggin, which are expressed in the posterior notochord and PSM. Thus, BMP signaling is restricted to the posterior tail bud, as indicated by phosphorylated SMAD. And they kind of show the seroimmunolocalization. They show even skip genes or EVE. This is the transcription factor EVE. It's one of these even skip genes that you see in Drosophila as well. These are adjacent to the HOX13 end of the HOX clusters, suggesting a role in posterior animal development. So this is the EVE and the BMP signaling working together. So one is involved in sort of setting up, morphogenesis of the tail, and it's a morphogen, and then even skipped is some sort of HOX-related gene that has a role in segmentation. Yeah.
This looks like a really good combination mechanical and chemical system at work right
Yeah.
here.
Yeah, I think so. I'm not sure the mechanical work, I wonder what they do in terms of mechanics. Let's take a look at this figure. So this just shows, some of the changes in some of these biomolecular factors and how they assay them. So they're looking at the expression of different things at the tail bud, looking at that. And this just shows like the different treatments here. So they have this administration of DMH1 and DMSO, and then EVE1. So you see that it's a transgenic EVE1. So I think they're overexpressing EVE1 here. And then this is a vehicle, I guess, DMSO. And then DMH1 is the perturbation. So you can see when you have the transgenic EVE1 plus the DMH1, you get this truncated looking tail. And you see actually the body's all scrunched up. When you have the transgenic EVE1, you see, like, I don't know, it kind of looks like it's maybe a little bit truncated, but it still looks like a zebrafish embryo. DMSO has a zebrafish embryo in full form. And then the perturbation DMH1 alone without the transgene, it's just kind of like the tail kind of is misshapen, if you can see that really
Yeah,
Clearly
that
couldn't.
scrunched one, I have an axolotl. It's kind of got a scrunched body like that.
Oh yeah.
You don't know.
Yeah.
Yeah. Anyways, yeah, I don't know if she can lay any eggs or not. And,
Yeah. Well, I mean, yeah, I don't know what the consequences of misdirected morphogenesis are. I mean, usually it's in that region, but sometimes there are other effects as well. So,
hmm,
yeah. All right. So that's that figure. Um, yeah, and then so they talk about cell motion. Cell motion in the tail organizer is aberrant in both DMH1 and transgene Eve1 plus DMH1 embryos. So whenever it's being perturbed by DMH1 in the wild type and in the transgene, cell tract straightness is reduced in both conditions, and the mean coefficient of variation of cell tract speed is increased in DMH1 embryos. So there are these differences in terms of how cells are moving around. Um, and so the directional velocity is reduced in the uh, tail organizer of DMH1 embryos. Cell flux through the tail organizer includes a medial domain comprised of both a dorsal to ventral flow and a medial to lateral flow that are segregated from flows on the lateral periphery that are predominantly posterior to anterior and ventral to dorsal. So these are flows that enable cell migration, and they're slowed down in these perturbed phenotypes. Um, these flows intermix after perturbation of tail organizer signaling, and the disruption of the dorsal to ventral flow likely obstructs cell flow into and through the tail organizer, particularly in the transgene Eve1 plus DMH1 embryos. So when you get this sort of transgene plus the perturbation, then that's really when you disrupt the mechanical flow of the system and you get this sort of phenotype that we see here, where it's really kind of pronounced. It's because
mhm.
this flow is restricted coming down the axis, the anterior to posterior axis. You were going to say something?
No, I was just looking at it. The most normal one is the uh, top left. But anyways, it's okay.
Yeah, so near.
Yeah, the first one there is the most normal looking.
Yeah. So this is G. This just shows like the different. I guess these are flow lines or something. Let's see what the uh, legend says. Full caption. All right. We're looking for G. Cell flow within the tail organizer, visualized by displaying only cell tracks with the greatest displacement from dorsal to ventral green, medial to lateral yellow, posterior to anterior red, and ventral to dorsal blue. So if we look at this figure here, we see that they're like looking at the cell tracks, but they're looking at their sort of displacement and anatomical space. So if you're, going in different directions and there's a greater displacement. So in other words, the displacement, I guess, well, the DMSO is the vehicle, so we can look at that and then compare with the other ones. So we can see that most of them are green and yellow, sort of here. And then on the peripheral regions, you have blue and red. And we said that those are um, blue is ventral to dorsal, red is posterior to anterior, dorsal to ventral is green, and then medial to lateral is yellow. So that's what we have for DMSO. And then if we look at DMH1, we have, it doesn't look like that. It looks, the shape is different of the cell tracks, but then you have more red and more blue, and you have less green and more apparent yellow, at least from the surface. With the transgene Eve1 plus DMSO, you see difference here as well. It doesn't look so much like um, it doesn't look so much like just the DMSO, but you get this difference in the tracks and their distribution. And then where it's really pronounced is in the trans-GV1 plus DMH1, where it doesn't, it's kind of looks disorganized, and there are a lot more red tracks and blue tracks. And then, you see some green tracks, but they're not really predominant. Like
Yeah,
they're,
that's messed up, the last one.
yeah, that's really messed up, I guess is the way to describe it. So, yeah, you can definitely see from these cell tracks the differences in the phenotype.
Okay. Well, I don't know where my other paper went. Um, oh, well, it's a long one, and it is more in line with my Tensegrity mechanics. So
Okay.
I I need to do I need to read it thoroughly.
Yeah.
I'm supposed to be writing a paper using it.
Okay. Does this have, like, is this mentioned Tensegrity directly, or is it just kind of like relevant
Yeah,
to?
I think so. And now
Oh, wow.
now I really want to look for it.
Yeah.
Of course I do. Oh, there it is. Oh, okay. I just have to get to the bottom of the pile. And the kids' book. Okay. Yeah, it says tensegrity structures and data-driven analysis for 3D cell mechanics.
Wow. Yeah.
Yeah, so that's right on, that's right on the money. And it says it's to be published in 2026. So this is a pre-print, I guess.
Okay. Yeah. Yeah.
It wouldn't do double-sided. It was a preprint.
Uh,
Anyway, yeah. So it's just they're making cells, 3D cells with the tensegrity structures, like a dodecahedron type structure, and then linking them together, which is not what I did, but this is, it's nice that they were able to do that, because linking tensegrity structures, as I've discovered, is sometimes impossible. They don't stick together, or they crash. I think they changed configuration too much for the finite element programs.
all right. So this is a computer methods and applied mechanics and engineering. So this is in January 2026. Apparently, they're already there in the future. This is computer methods and applied mechanics and engineering. So this paper kind of goes over different tensegrity structures, and then they're analyzing this, and they're looking at 3D cell mechanics. So they talk about the cytoskeleton playing an important role in many cell functions. Given the similarities between the mechanical behavior of tensegrity structures and the cytoskeleton, many studies have proposed different tensegrity-based models for simulating cell mechanics. So then, of course, they talk about the low symmetry of most tensegrity units and how this has hindered the analysis of realistic 3D structures. They're talking about taking the tensegrity units that we've talked about in the meetings past and how you can combine those, because obviously you need to combine them to get this, to make this useful to tissues. As a result, tensegrity-based modeling in cell mechanics has been mainly focused on single cells or monolayers. So in this paper, we propose a 3D tensegrity model based on the finite element method for simulating 3D cell mechanics. We show that that proposed model not only captures the nonlinearity of a single cell and an indentation test and a monolayer and stretch test, but also the non-uniform stress distribution in multicellular spheroids upon non-uniform pre-stress design. Furthermore, we introduce a multi-scale data-driven framework for cellular mechanics to optimize the computation, thus paving the way for modeling the mechanobiology of large cellular assemblies such as organs. And
And this is interior cells. It's not epithelial tissue or endothelial tissue. It's the interior cells,
okay, yeah,
which is nice. Yeah, this is.
yeah. So yeah, they talk about how the cytoskeleton senses and transmits forces to interact with the adjacent cells and microenvironment. This orchestrates cell shape changes, polarity, and motility. An equilibrium in forces is maintained at the cellular level, balancing internal mechanical forces exerted by the contractile actin filaments, resisting microtubules and compression, and external forces from the extracellular matrix. This state of pre-existing stress in the CSK is referred to as pre-stress. Multiple studies have reported the central role of pre-stress in cellular mechanotransduction and mechanobiology. Okay, and then they get at sort of the traditional computational efforts, sort of, continuum mechanics that are used, describing the cell as an elastic, viscous, or viscoelastic medium. This is assuming the cell is composed of materials with certain continuum material properties. So, you have to assume that the cell isn't just a sphere, but like has these different properties all acting kind of at the same time. So, you have in, say, like a typical cortical shell liquid core model, where you have the outside, which is the shell, and then the liquid core in the middle. We assume single or several layers of cortex with surface tension, with the cell interior modeled as Newtonian fluid with certain viscosity. So you have this sort of composite model where, different parts of the cell have different properties. You have this certain behavior of Newtonian fluid with viscosity. Other continuum models simulate the whole cell as a homogeneous solid. So some models just kind of, treat the whole cell as a single entity, and that's how they kind of work on it. Some such continuum models are good approximations for a range of experimental observations. However, as experimental conditions become complex, constitutive models require additional parameters that are essentially empirical and often lack in physical meaning. So, if you use a homogeneous model, it's basically just kind of like, you might be able to map it to a single measurement. But once you start measuring more parameters in the cell, then those become less useful, especially when you consider complex physical interactions. So we can't, it's hard to consider subcellular processes such as protein level activities. And to get around that, sometimes people used averaged values. So, you can use a mean field model or an averaged model of things like protein level or other types of parameters. But the problem then, again, is this aspect of what individual cells are doing. And so if you're looking at a cell sheet where there are a bunch of physical interactions, each cell is going to have specific values for those things, and you can't average them together necessarily. You're smearing out a bunch of important information about the mechanics of the cells in that structure, and you're smearing out the sort of the biological properties that are interesting. And so then they talk about these individual cell based approaches where we have cells treated as individual entities. They talk about lattice based and off lattice models. Um, the former, which are lattice based, track cells along rigid grids. So you might have like a fixed lattice cellular automata or a cellular POTS model, which they use in CompuCell 3D. So that's
Mm-hmm.
the lattice based model. And then the off lattice based model is where they don't have a reference grid. And so cells are just basically tracked as centers of mass. So this is like the point clouds that we use in cell tracking data. We track the nuclei, we track the centroid, and we don't worry about a reference grid. We just maybe have a coordinate system, and those centers of mass can interact in different ways. and we can measure them. The benefit, of course, of having a rigid grid is that you can define sort of the relative position more clearly. So there are pros and cons to those. Um, and then, of course, we have vertex dynamics models where additional biochemical rules can be employed to study more advanced cell behaviors, such as cell rearrangements and oscillations. And so they kind of go through this review, and then they talk about the tensegrity principle, which is observed in various natural systems across scales, from the kinematics of human and animal locomotion down to the molecular architecture of spider silk fibers. And so within the CSK, which is the cytoskeleton, the force balance between contractile actin filaments and compression supporting structures such as microtubules is reminiscent of the self-equilibrium of tensegrity structures. So they're talking about like mechanical tensegrity structures, um, such as geodesic domes and things like that. And so there's this idea of opposing forces balancing each other out. And so that's what they're kind of trying to get at here. So they're talking about pre-stress and how cell stiffness in individual cells can scale with that pre-stress. In accordance with prediction from tensegrity models, these factors render tensegrity a conceptually simple but relevant idea for simulating cell mechanics. And then they talk
Yeah,
about
it's not easy to simulate it.
no. Yeah, it's going to be hard because you have all these different factors that go into it, and they're important for, I take it, for determining things like pre-stresses and like the opposing balances of forces, things like that.
Well, and it's non what is it non determinant because it's got a basically a chaotic structure, which is a double pendulum. Like if you have two rods or two compression elements together, then and it's like it works like a pendulum, and you don't know whether it's going to go this way or that way because you're swinging this one, and this one also swings. So it tends to be a chaotic system, and it's attached to the elastic members or the string or tension members. And so the thing is indeterminate because it's got this chaotic system. If you've got more than one compression element attached to the strings. So it's so it doesn't work with finite element analysis all that well because it's basically indeterminate. Like it can go one way or the other, and you don't know which it's going to do really, until you put that last little minute force on it in the finite element analysis. You put tiny little forces on your model, and then all of a sudden, in a tensegrity, it will change shape. Boom. Yeah.
So small forces kind of result in a configurational change.
Yes.
So if we go back to the paper, they propose that a 3D multicellular tensegrity model capable of simulating cells in 1, 2, and 3 dimensions, or what is kind of the answer to a lot of these modeling problems, and investigate the response of such structure under different loading conditions. They base their tensegrity modeling framework on a finite element method in finite or large deformation settings and allows controlling initial pre-stresses. As with any finite element model, the computational cost becomes exceedingly expensive as the number of elements increases. Therefore, to reduce computational costs, we further perform simulations using a multi-scale data-driven approach in concert with homogenization techniques. So this is where they're using averages and not accounting for like every single nuance of the cell, and they're just using that but they're using this as a way to sort of add data to this model so that they don't have to simulate everything from first principles. So, there are kind of decisions you're making there to sort of balance computational costs with realism. And that's, it's just kind of where they are with this. Data-driven computing enables calculations directly from a material data set while satisfying pertinent constraints and conservation laws, thus bypassing the empirical material modeling step in the conventional finite element model. So they kind of go through the tensegrity formulation. They simulate a single cell, then a monolayer, then a multicellular spheroid. So they kind of build up to the multicellular case with geometry. So you have like kind of the single cell, which is the point, the monolayer, which is this, one-dimensional or two-dimensional structure, and then multicellular, which is the three-dimensional structure, the spheroid. And then this is compared with experimental work. They kind of go through the 3D monolayer simulations, and then the solutions are then benchmarked against the tensegrity-based direct numerical simulations. So they kind of go through their model of tensegrity, where it's composed of a pin-connected tensile and compressive set of elements. They adopt the modeling framework of Ma et al., which is this reference here, and they just summarize it below. They use Cartesian coordinates to characterize the node. It's X, Y, Z, and then you have this matrix, and then you have your node number, which you can identify single nodes in this global nodal coordinate matrix, which you construct here as large N. And then in a single vector formulation, the nodal coordinate matrix reads small n, and then the elements here. So you can build a coordinate matrix as a single vector. The connectivity matrix then represents the topology of the entire tensegrity structure, which consists of M sub B compressive bars and N sub S tensile strings. So you have these compressive bars and tensile strings as the edges of the network. The nodes of the network then are these nodes that they define up here in this matrix large N and this vector small n. So then we have this this conditional here, the stack where you have basically you have like the compressive bars and the tensile strings, and that's combined into this M here, which is its corresponding kth row and ith column of entry CL. So you have this conditional where you have 1, -1, and 0. So basically you're classifying something where the L value is either I or J, otherwise it's 0. So this kind of gives us a way of classifying these different conditions. And then each element has a length, which is here. The length is either I or J, calculated here. Accordingly, the global structure length vector is assembled here. We define the global area vector and modulus vector given by capital A and capital E. In each element K, the axial stress increment, which is here, is given in terms of the axial strain increment, which is here. The general tensegrity framework is equipped to handle elastic or plastic materials. In this study, we assume the members are elastic and have constant cross sections and moduli, focusing on structural behavior under moderate strains without introducing additional complexity from nonlinear material behavior. So we have this internal force vector here, where L0 is the rest length vector before the application of prestress. So you have this rest length vector, and then you're adding prestress. You're adjusting L0 and L, which can achieve different levels of prestress in the structure. Okay, and then you have force density vector Q, which reads here. reflecting the geometric nonlinearity through the current length vector L. Okay, so then they go through some more, they go through statics, and then they have an identity matrix, and that's supposed to give you this structure at equilibrium. And then you have, since k is dependent on nodal coordinates n, the left-hand side of equation 13, which is here, is expanded using the chain rule. And then from equation 10, which is up here, which is the force density vector, we can show that the partial derivative of q with respect to n is given by this equation, which is now we're getting into differential equations where this AEQ is this value.
Yes, linear algebra on top of advanced calculus.
Yeah, yeah. So we're getting deep into linear algebra and then moving to calculus. So we can,
and then throw it into calculus. Yes.
yeah, so we can characterize the dynamics, right? Yeah, I think, yeah, maybe that's well, I mean, it makes sense, right? Like, because you're working out the network structure and then you have to simulate the dynamics.
Yes, it makes sense that they're doing this. It's just, I think you lost me a couple of lines back, but yeah, it's okay.
It's okay.
I wish I could get a complete course on this stuff.
Yeah.
Somewhere.
Yeah, it's a lot though that you're modeling because you're kind of going through and you're saying, okay, here's the structure. We have all these different components. We have to balance them out. Then we also have to have like we can do the 2D case and the 3D case, and then we'll just kind of do all that, then do the dynamics, and then it's yeah, you're tracking a lot of things through here. Well, anyways, so then they go into sort of getting to the states of self-stress and the sort of the structure, structural loads and things like that. And then basically you have to sort of introduce the geometry then. And that's here. And then there's this 3D tessellation that they talk about, which is where tensegrity structures remain in self-equilibrium, which requires special arrangements of the elements. And so now we have to sort of think about form finding of eligible self-stabilizing tensegrity configurations. So this is where we have to find the eligible configurations that can be determined as stable. So like you can, it's like kind of like protein folding, where you have all these configurations, this huge configurational space. Some proteins are functional because of their configuration, and some are not. And so such it is with like tensegrity structures. If the structure sometimes it's not viable, and sometimes it's viable and stable, and that's what we want to find. So you can imagine this is a big computational problem because you can imagine with protein folding it's huge in terms of like simulating just a little bit of the protein folding process. I mean, this has been the problem for years. They've had to kind of simulate just a little bit of protein folding, and that's taken up a lot of computational power because there's so many configurations that it can take and that have to be evaluated. So, I imagine with these tensegrity structures, you can't get a very, long sequence of dynamics to really kind of evaluate. So this is really going to be just kind of like, I don't know what their time scale is here, but it's going to be hard. It's going to be a hard computational problem. So this is the image here where they talk about, see if I can open this up. They show these structures. So you have these networks where you have the compressive elements in black, the tensile elements in red, and then these geometric shapes within that structure. And then if you look at this you can look at this high-res image, you have sort of the symmetries. So you have the original structure, which is where you have the sort of the top and the bottom and the left and the right defined. And then you have the mirrored structure, so you can look at the mirror image. And then this shows the symmetries. So you have the original, you have a mirrored symmetry on the y-axis, a mirrored symmetry on the x-axis, and then on the z-axis you have like basically rotating So you have these mirror images.
Hmm.
Yeah.
I'd like to know why they have solid filled in parts, or why they went that way.
I don't know. So you're talking about the interstitial, like the yellow and the gray.
And the gray, yeah.
Yeah, I don't
Why?
know. Yeah.
Why that? Why did you do that?
I think it's
What's
probably to demonstrate kind of like the stable surfaces or like the surfaces on the network, but also show the elements underneath.
oh, maybe so you can see it, or
Yeah,
is it
yeah. I
part
mean,
of the
like
structure?
illustrating, like if you just said that this forms like a shape, you'd say, well, there are a bunch of lines there.
Yeah, yeah. It looks like a bunch of lines. That's what my thesis was about. Lines all over the place,
Yeah.
and it was sometimes hard to keep them straight.
Yeah, yeah. So then this is how, so this is like the neighborhood of
Uh,
connections. So if we think
okay,
about
so
this as a
there
network,
they don't have things colored in. They they just have the structure. Okay,
right?
all right.
Yeah, and then this is like kind of the more typical tensegrity you'd see with like, um, where you have the different, like basically, I guess these are cells that are next to one another.
Yeah, but
Yeah,
you notice there's nodes there with what is it? 6 black lines coming out of it. I don't know if you can't see, but um, there's more than one black line meeting.
okay. Yeah,
Um,
yeah,
so that means it's um, non-determinate tensegrity structure. Yeah.
okay.
Anyways,
All right. So it's like when you have multiple lines.
yeah, yeah. When you have multiple bars, which are the compression elements, then it's indeterminate.
Okay.
It has been labeled indeterminate. Yeah.
Because you
I
have
can show
multiple.
you multiple bars, and it acts like I said, a multiple pendulum situation.
Okay.
Yeah, and I can show you the chart. This wasn't invented by me. This was it's in Gann's book, which is hard to follow, but and misleading. Oh, you just do this. Yeah, right.
Yeah.
Mm-hmm.
Yeah. Okay. Yeah. So that's the paper. Yeah. Thanks for showing us these papers. Very interesting to follow through from like networks to then tensegrities.
Well, a tensegrity is a network,
Yeah. Yeah.
and yeah, and it changes like um, during I don't know whatever a cell is doing, but at a moment in time you'll have a certain tensegrity structure in a cell. It'll be stiffened in one direction and in tension in another direction, and the whole thing holds up, but um, you can probably call it a tensegrity structure.
Yeah.
But the analysis of cells mechanical networks is in its infancy, I would say. They
Yeah.
haven't, there's not much out there on that. And to check it for a tensegrity structure, to see if it is a tensegrity structure.
Yeah.
So I'm interested in mechanical um, networks in cells.
Yeah.
Interested to see that because um, basically you have some parts in tension and some in compression, and that forms a tensegrity structure. So
Yeah.
anyway, yeah, thanks for doing this with me.
Oh, no problem. Yeah. All right.
You
Well.
know, I have no one to talk to about this.
Oh, yeah.
The mechanical engineer that I live with here, he goes Tensegrity, yuck, and walks away.
Oh.
Conventional mechanics, fine.
Yeah.
Yeah, he's actually a fairly good mechanical design engineer, but he doesn't like tensegrity structures.
No.
No. Anyway.
Right.
Yeah.
All right. Well, I think that's all for today. Yeah. Thanks again for the session.
Okay.
All
All
right.
good.
Have a good week.
All right. You too.
Talk to you later.
All right.
Bye.
Bye.
All right. So, several meetings ago, probably this summer, we talked about this book, Laws of the Game, by Manfred Eigen and Raphael Winkler. And this is a book that talks about game theory in biochemistry and physics, and it talks a little bit about their application on like cellular automata or in computation. So Manfred Eigen, of course, was the pioneer of things like the hypercycle, but he also had this interesting game theory where you can play these games that mimic things in biochemistry or in physics, and they can have you can have these different strategies for playing them and all of that. And so in this book, one of the types of games he talks about are these bead games, where you have beads on lattices. Like we talked about in the meeting, these lattice methods that people use for modeling. And you can model different types of scenarios on that kind of board. So it's not quite like the game Go, but it's a similar principle. And so I think it was Hussein who mentioned that chess is a stupid way to think about game theory, and maybe these kind of bead games are more appropriate for things like biochemistry, which I tend to agree with because again, chess is a specific type of strategy, and these bead games are much more useful for things like biochemistry, especially when you have competition and you have different types of processes, dynamical processes you want to understand and replicate. So the other reason I bring this up is because I found this website. This is David Ellerman, and I guess he's some sort of polymath. He does a lot of stuff with like economics and other types of law applications. And he also does like quantum mechanics and mathematics. And he has this set of Excel spreadsheets, which are a little bit archaic way to do this, but he's modeled some of the games and the laws of the game in these spreadsheets. And I wanted to look at these spreadsheets and go over some of the games that he's taken out of the book. So the book itself is, proposes these bead games. It proposes different types of strategies for playing them. And then he's taken some of those things out of the book and run some simulations and spreadsheets. So one of the things he talks about is the Ehrenfest-Earn model. And this is there are a couple of versions of this. This is with the random initial state. So if you go to the book and you look up Ehrenfest-Earn model, you'll find that, and this is a screenshot of the spreadsheet. So I have the spreadsheet in sort of Google Sheets now. And but this just shows the structure and how this is calculated. So there are equations in these cells, and you can have an input matrix here, find the sums, and then this just shows the distribution or the ensemble that you get. So a couple of notes on this: the initial distributions of zeros and ones is random. So basically we have this matrix of zeros and ones that come from these inputs on each axis, and then you have the sums down here, and then you have zeros represented by an empty cell and ones represented by these black boxes. And so you have this distribution of black boxes as a sort of ensemble that is the result of this random initial condition. So the initial distributions of zeros and ones is random, and then you play the game and you get this, mixing that occurs. So you get this random distribution of zeros and ones, as you can see. Each iteration in a cell in the 16 by 16 array, which is the size of our grid here, is randomly selected. The entries can be 0 or 1. When a cell is selected, its entry flips to the other value. Since the most populous value tends to be selected and flipped, it is negative feedback. The equilibrium towards which it tends to is where you have 256 divided by 2 equals 128. So, in other words, like half of the cells will be zeros, half of the zeros will be 1, and, that should happen over time. So, what you have here is you have this initial distribution, which is random in nature. These black squares are randomly distributed. Then we randomly select cells and we flip the values. And we keep doing this over time as we select cells. And then what happens is that the most populous values tend to get selected and flipped. So when the most populous values get flipped, it's negative feedback, which reinforces order. And so this is a nice sort of way to show how order can emerge from randomness. This is the Ehrenfest urn where we have a uniform initial condition, or where we have an all black initial state. And so again, we have an initial state that's all black, and then we play the game and we get to the point here where this array occurs. And so initially each cell is black, each ball in our... So what we're doing with a random process, the reason they call it an urn, is because there's this whole class of problems in statistics called urn problems. So you have different urn models proposed by different people. You can model different types of sampling in this way. So you have this urn, which is like a black box, and you have a bunch of balls in the urn. And the idea is you reach into the urn and you pull out a ball. It's either black or white, in this case 0 or 1. And you don't see what you're pulling out, so it's a blind process, blind with respect to selection. And you're pulling out a ball and you're putting it on the table. So the idea is if you reach into the urn enough times and you pull out balls, the distribution you'll get is random in nature, and you should get a random distribution. That's the idea. Now, in this case, so in the last case, that was a random initial distribution. We're drawing balls out. We have an equal number of zeros and ones, and we're just drawing them out at random and placing them on the board. In this case, we have a uniform initial condition, which is that every ball in that urn is black, and so when we reach out and we put something on the board, it's always a black ball, and all of our cells then have black balls in them. And then once we populate our grid, then we evolve our grid. So each iteration in a cell in the 16 by 16 array, so we have 256 spots, is randomly selected. So the entries can be 0 or 1. So we start all with a uniform state, and then we start to iterate on that by flipping the values to things that are not black balls. When a cell is selected, its entry flips to the other value, so we select something, it flips. And this happens over a number of iterations. Since the most populous value tends to get selected and flipped, again, it is negative feedback. And so then our equilibrium tends over time to be where we have half of our grid being white balls and half being black balls. And we end up in the same place that we do with the first example, where I showed you that it was a random initial condition. In this case, it's a uniform initial condition, and because of the way the game was played, we end up in the same place. Okay, this is the bead game, which was from page 52 in Laws of the Game. So this is selection. So this is where we have our 1,000 iterations of this game. We have this lookup table where we have this set of 1s and 3s. And so this is an 8 by 8 matrix. And then now we have our relative changes here over 1,000 iterations. So in this game, on each iteration, a square is randomly selected. The value is doubled. and its content is then assigned to the second randomly selected square. There are 4 possible contents, 0, 1, 2, and 3. So what happens here is we have any one of these values can be 0, 1, 2, or 3. In this case, we have 1s and 3s only. And each of these are represented by, this is from a deck of cards where you have clubs, diamonds, hearts, and spades. And then, each state from 0 to 3 is represented by one of these symbols. So you can get a sense of what the contents are by that. So you have this ensemble with different distributions of these symbols. On the first iteration, the contents of the squares are randomly assigned. Eventually, one suit will tend to dominate. So the idea here is that we again have this case where we randomly distribute these symbols. We can use an ERN model to draw out, we can have a number of these symbols evenly distributed. We can place them on the board, but over time, as this game progresses, you end up with one symbol that predominates the board. And so in this case, I don't think that's the case in this iteration, but for later iterations, say like, for example, we look at this, we have all 4 symbols evenly represented on the board. We keep playing the game as we see here in the rules, and then we end up over time with enough iterations getting a single symbol that predominates. So you might have most of the board filled with hearts, or all of the board filled with hearts. So eventually one suit will tend to dominate. One can keep score by watching the relative changes relative to an initial random assignment. So you can actually score this and see the dynamics of this change. Okay, so that's that's the bead game in selection. This is going back to the Ehrenfest model with cooperative phenomena instead of what we showed before with an initial condition. This is where we have this matrix, and we have 600 iterations. We have a matrix of 0s and 1s with the sum down here, how many 1s you have in each row, or in each column. We have our neighborhood sums here, and then we have our board where we have red squares and blanks. And so that's what our board looks like. It looks different than the other boards. And why is that? So let's take a look at the instructions. So the entries in the 16 by 16 array can be 0, blank, or 1 red, these red squares. In each iteration, the entry in a cell flips to the other value with a probability of proportional to the number of 8 neighbors equal to the other value. So in this case, we have these neighborhoods of 8 cells surrounding an initial cell. So this cell here is going to have 8 neighbors. It's going to be every cell that surrounds it. 6 on the top, 6 on the bottom, and 2 on each side. That neighborhood of 9 cells then will shift. So, this cell will have a neighborhood of 8 neighbors. And this cell will have a neighborhood of 8 neighbors. And this cell will now be a neighbor of this cell. So you can see the parallelism where you move across the board, and each cell can either be sort of in the middle of a neighborhood or in multiple neighborhoods. And so that's what they mean when they talk about neighbors. And so we also see up here where we talk about our neighborhood sums, we have our neighborhoods defined and the sum within that neighborhood. So what we're going to do is we're going to start with our 16 by 16 array. We're going to have multiple initial starting points. We're going to flip the entries in a cell to some other value with a probability proportion to the number of the 8 neighbors equal to the other value. So when we have a majority of cells that are blank, that central cell will then become blank. This is very similar to what we do in cellular automata in neighborhoods, in more neighborhoods. And so then the initial distribution of 0s and 1s is random. So we can have an initial distribution that's random. We can have this update rule, and we can end up with, this sort of order that emerges. A higher stabilizer value makes it harder to flip a cell's value. So we can have stabilizer values that can change the dynamics of this game and make it harder to actually flip cells. Because sometimes there's this tendency in, for clusters of blank areas to be reinforced through this kind of a process. Sometimes it's easy for the whole simulation to converge on one state or another through this process, but there's stabilizer values that we can use as well. Okay, that's just for this instructions. Okay, this model was motivated by the description given on pages 71 to 72 in Laws of the Game. So if you're interested in following up on this and playing with the spreadsheets, they have this set of eigensheets. So this is kind of like for these different parts of Laws of the Game. So we have board game, bead game equilibrium. This is where you have an all black starting state. Bead game equilibrium, where you have a random start. There is this bead game once and for all, and I didn't talk about that yet. And so the once and for all game is a variation on these games, the bead game, but it has a distinct strategy. And that strategy is to try to have like kind of a solution that is a winning strategy. So in other words, the dynamics of a lot of the games in these, the dynamics of a lot of these bead games tend to lead you to sort of some sort of equilibrium state. It isn't necessarily where you have a game where you have all one state or all the other state. They tend to fluctuate with different internal patterns. But in once and for all, the goal of the game, the object of the game is to dominate everything, to finish off your opponent, and to become the dominant state. And so once and for all is sort of a way to model small-scale catastrophes. These kind of takeovers of one state or another is a small-scale catastrophe. And what I mean by small-scale catastrophe is to go to Renee Tom's catastrophe theory and think about how catastrophes operate. In other words, you have these sort of large-scale changes that occur that reorder a system. And that's kind of what you want to do with this once and for all strategy. And so the idea is that you have this, basically this internal mechanism in the game, or these strategies that play upon fluctuations in the dynamics of the game. So as there are changes happening within our matrix, we try to seize on those fluctuations and amplify them. And so there's an internal mechanism within this game strategy that's employed that responds to fluctuations in an exaggerated manner. So that means that, in terms of responding to these fluctuations, you put a lot of resources into sort of dominating the game. And so through these kinds of strategies, we can actually examine these games and look at the precursors of homogeneous states. So, if we see this in a system, if we see a catastrophe, like where we see an extinction of a species in a food web, we can actually go back and look at what the precursors were. What were the sort of dynamics of competition that led up to this problem? Or maybe there was an external perturbation. So what was, maybe that external perturbation didn't do the job by itself. Maybe there were other dynamics within the competition structure of that food web that led to that extinction event. And so once and for all allows us to look at those kinds of dynamics. And so once again, if you're interested in downloading these eigensheets, you have the bead game, and you have different versions of the bead game, and then you have the Ehrenfest model with cooperative effects. And those all kind of point to different pages in the book. And then finally, if you're interested in some of the other things that he's done. You have Kaufman sheets. So he's also done this for the book At Home in the Universe by Stuart Kauffman. These are modeling the Boolean networks and fitness landscapes that Kaufman talks about in that book. He's also implemented simple genetic algorithms, and then, the game of life, different iteration counters, looking at the glider gun within the game of life. He's done some epidemic modeling, some simple genetic algorithms here from John Holland's book, The Hidden Order. And then, of course, Polya's City. So I talked about Polya's Urn, or I talked about Urn models. And Polya's Urn is a version of an Urn model where we do sampling with replacement. In other words, we put a bunch of balls in the urn, and then we sample balls out of the urn. And as we sample balls and put them on the table, we get new balls in its place, and it's according to the original distribution of balls in the urn. So we're always drawing from a fixed probability. And so we can use these kinds of things in Bayesian modeling, where we have this process where we're gaining information and we can adjust the probability of that urn. We can replace the number of balls in that urn. So we have a continuous sort of distribution, and we can model things fluidly like that. And so this just kind of goes through Polya's urn and talks about sampling with replacement and how that works on a grid. So he plays this out on a grid. So Polya's City is a circle or ring of neighborhoods. When a location hits 0, it goes broke. To the neighborhood effect, a location that has gone broke can again go positive if it has a non-zero neighbor, and then it is possible for the next broke neighbor to go positive, and so forth. Okay, so this is basically where you have these grids with sampling with replacement, and you get this structure across space, and he calls it Polya's City. Very interesting stuff.